Hey, Stu! Happy Halloween! Wait, gotta get back in character. So, you've come late to lab once again, and you've entered into my laboratory of your own free will. This means you cannot escape the horror that awaits you as you become one of the children of the genetic corn. Seriously, I just need a minute to put on my lab coat and we'll be ready to get started with the lab.
Yo, Stu, what's up, man?
What up, Ben?
Hey Stu, what's up?
Traits like hair and eye color, flower color, leaf shape, and fruit variety are all inherited on genes which lie on chromosomes. During meiosis and sexual reproduction, each offspring gets half of its genes from each parent. We can predict the outcome of a cross using Punnett squares and probability.
Well, welcome back. I've defanged and I'm feeling much better now. So we're moving on to the genetic corn lab, which comes in this handy dandy kit. To do this lab, we do need some paper towels, a way to make them moist. Opening the kit, we've got some wet erase markers, our lab sheets, teacher's guide, and corn. I know it's a bit of a shock that the corn lab would actually contain corn, but it's a corny lab, what can I say? So we've got C corn, we've got A corn, and B corn. We don't need the C corn for now. A little extra paper for us to scratch on. So let's get a little bit of background. Believe it or not, corn is a grass. Corn is one of the weird grasses that the Native Americans learned when two grasses grew together, their kernels grew really large and they were able to eat them. So it was the Native Americans who first started breeding ZMAs to make a corn that produced a kernel that was good to eat. Now, back Thanksgiving kind of time, their corn was very hard and very flinty, and all they could really do was make a paste or a succotash out of it, but it kept them and the Pilgrims going for a really long time, and they learned a lot about genetics, and we're not even going to go, to the Mayans and what they did with corn. But in this case, this is corn that has been selectively and very specially bred in order to prove Mendel's ratios and Mendelian principles. So these are two test crosses, and eventually we'll cross this corn and this corn for another corn that's over there. But let's get to the idea of what we've tried to do here. So we want to know, was Mendel right about the law of dominance? Law of dominance say that dominant traits will be seen over recessive traits. Dominant things are dominant. Law of segregation, and that is that alleles separate during meiosis and you get a copy from mom and a copy from dad. The second part in the dihybrid cross will be the law of independent assortment, and that is that different traits assort independently during mitosis. So we've got our kit from Carolina Biological. You missed the due date, but we'll get there. So let's look at the first cross here, cross A. So we have two possibles here. This is purple, this is yellow. Would you agree that purple is dominant over yellow? Yes. Since we're starting with true breeding homozygous traits, this is going to be big P, and this is going to be little p because it's recessive. So here on the lab sheet, so the dominant would be purple, RPLE. Recessive is going to be yellow. And we would expect this first cross all to be, let's see, big P, little p. We're expecting these to be heterozygous, and we're going to have 100% purple and 100% purple. If we were to do a Punnett square, and a Punnett square is to predict the probability of a cross, we would put big P and big P, little p and little p, then we drop them into the boxes, big P, little p, big P, little p, big P, little p, big P, little p, and that explains what we get right here. So both of these are big P, little p. Now we have a hybrid, and we would call this hybrid homozygous dominant. Now, if you were to cross this and this, or shall we say this and this together, you're going to get this. Now, as we go to describe this corn in the lab, what does this ear look like? Would you agree that this is purple with some yellow? Purple with some yellow? Yes. Okay. Purple with some yellow. And again, the parents here are going to be big and little and big and little because that's how the alleles are going to sort. So we're going to get a big P, big P, big P, little p, big P, little p, little p, little p. Genotypically, we have one homozygous dominant to two heterozygous dominants. That means one big, one little, and one homozygous recessive. But the law of dominance says that purple is dominant over yellow. So we have 1, 2, 3 purple to 1 yellow. Ta-da. So we need to count it so that we can prove this ratio. So we're really looking at 3 purple to 1 yellow. And that's, a 3-to-1 ratio. So when we count this corn, we should count how many purple, how many yellow, and then divide both of them by the smallest one, and we'll see. So what I suggest is you make tally marks. So we're going to call this purple, this yellow, and you're just going to make tally marks 1, 2, 3, 4, 5, and I'm going to count them off to you. So you take that, I'll take this. And yes, we count every kernel that we can see. So 1, 2, 3, 4, 5 purples, 1 yellow. Yellow, yellow, purple, 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 yellow, purple, purple, yellow, purple, purple, and I'm back all the way around the corn. Yay! We didn't bother to count the ones underneath the label, and we certainly didn't want to disturb the plastic. But now we have some numbers that we can work with. So to clean this off, it's fairly easy. It's a wet erase marker, and it's ready to go for the next person who needs to count it. Of course, in the kit, there is enough for 8 groups to be counting at a time, but that's good to go for next time. Looking at about your numbers that we have together there, it looks like we roughly have, by your count, about 209-ish purple, so 209 to 78, I think, no, 76, 76. That brings us to a ratio of, so 209 divided by 76 plus 76 divided by 76. Divide both counts by the smaller count. Should be a number, show the math here. That's our ratio. That's going to give us a ratio of 2.7 to 1. Now remember, we projected a 3.3 to 1 ratio. Now granted, not every kernel of corn has the same number of kernels on it, so that's pretty good. And that actually jives with some of the classes data. If you look up there on the front board under purple versus yellow, you've got my first block's data and your fourth block's data, and what their ratios worked out to be. That's purple versus yellow, which is the middle board. But let's move on to our second cross. So that's A. Looking at cross B, we're looking here at sweet versus starchy, or sweet versus wrinkled. So the dominant trait is again sweet, and we'll call that wrinkled. So this is going to be, why don't we go big S, and then we'll call that little s. About the naming conventions, okay, you know this is the homozygous recessive, therefore it needs 2 letters because it got both of that from its parents as a pure breeding strain. We need 2 copies, one from mom, one from dad. Again, these are both going to be big S, little s, big S, little s. That brings us to this cross right here. So filling out the chart again, we want to show the parental cross, so big S, little s, little s, big S, little s, big S, little s, big S, little s, big S, little s. It's going to give us 100% because they're all heterozygous dominant. For the F1 generation here, again, big S, little s, big S, little s, big S, big S, big S, little s, big S, little s, little s, little s. Again, 1 homozygous dominant, 2 heterozygous dominant, and 1 heterozygous recessive. Again, sweet, 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 wrinkled. So what does it look like? It's mostly sweet with some wrinkled. And we should expect a 1 to 2 to 1 versus a 3 to 1. So now we're going to do just what we did before, and that we're going to count all of the kernels. Now, not to let you not have the fun, do you want to count and I'll tally this time? Sweet. Finally done. So it looks like 111 of the sweet to 45 of the wrinkled. Again, dividing both the counts by the smaller count, we get a ratio of 2.45 to 1. We're still looking for the 3 to 1, and we're not that far off, and corn's kind of stubby, not many ears there. There are 8 different ears in the kit, so hopefully over a larger set of data, you're going to get closer to that. But having dealt with A and B, if we can look at these two families. What if A and B had a baby? We crossed A and B, we're going to get the C corn. But in all of these cases, we are looking at monohybrid crosses, where we're crossing sweet versus wrinkled or purple versus yellow. One set of alleles, one trait that we're looking at. When we are now mixing kernel color with kernel texture, and now that means we need to do a dihybrid cross. A dihybrid cross. So what does the ear look like? It's a hot mess. You've got purple and yellow, sweet and wrinkled. The possible genes that A can give you is a big P or a little p. What this corn could give you would be big S or little s. So what we need to do now is predict our dihybrid cross. Here we're getting into independent assortment and what are the four different types that we're looking at. So if we're crossing big P, big S, little s, we're going to generate the gametes through meiosis, we're going to have to foil. You remember foil from math. First, outer, inner, last. So as we generate the parents, we'll go with first, so that's going to be big P, big S. Outer, big P, little s. Inner, little p, big S. Last, little p, little s. And that's going to be the possible genotype for both parents as we go here. Let me attach that. So, drop my lines. Ooh, I'm uneven. Big P, big S. Big P, little s. Little p, big S. Little p, little s. Big P, big S. We needed to do the foil because we've got to independently assort the traits from both parents into each gamete. And that means we're doing two now. So what we do now is fill in all of the letters in each of the boxes. So big P, big S, big S. So, we've now predicted what our possible offspring are going to be. Now, you know in Punnett squares that a Punnett doesn't mean that you're going to have 4 kids. It means this is the probability out of 100 of the likely genes of your offspring. So, when we double it and go dihybrid, then we have to go. Now, the genotypic ratio is right out because we've got so many different possible gene combinations. We don't have a 1 to 2 to 1 that we can just suavely throw out there. Instead, we're not going to worry about that. We do need to look at the phenotypic ratio, and we need to look at what possibilities we have. So let me grab a green and a red to go with this blue. So Mendel predicted for a dihybrid cross 4 different phenotypes. And he predicted that they would be in a 9 to 3 to 3 to 1 ratio between the two. So let's see if he's right. So if you've got big P and big S, that means purple and sweet. So how many purple and sweets are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's our 9. Purple and sweet. We should have 3 yellow and sweet. 3 yellow, or actually, 3 yellow and sweet. Those are going to be little p's and big S's. So yellow and sweet, yellow and sweet, yellow and sweet. So this is purple, big P, big S. This is little p and big S. We should also have 3 that are purple and wrinkled. Purple and wrinkled would be this one, that one, and that one. So we're right again. And then 1 that is the double recessive. Right here, this little guy. 1 double recessive as our 1. So that means that we are looking for a tally of 4 different traits. We need to know how many purple and sweet, how many yellow and sweet, how many purple and wrinkled, and finally how many yellow and wrinkled. So that means we are looking for 4 different possible chances or types of offspring here. So I see some yellow wrinkled, and I see purple wrinkled, and I see regular yellow sweet, and I see regular purple sweet. So the next thing we can count is just those on a tally sheet. So, do you want to tally or do you want to mark? So you just want to mark this time? Okay. So that probably took a little longer than you expected, and it takes its toll on the fingers as well, but at least it's a wet erase, and it's easy to clean up, and it's ready to go for the next person who needs to come make up lab or comes to lab. Looking at our final tallies, we wound up with a ratio, instead of precisely 9 to 3 to 3 to 1, we came out with something that was 9 to 2 to 2.6 to 1, which is really, really close to Mendel's dihybrid ratio. But we just counted 1 ear of corn. To get to that magic ratio, we're going to have to count a lot more corn. A lot more corn. In the absence of counting a field of corn, we have gotten close enough to prove that it's actually right, that we're close enough to that. What's more than that, we could do statistical tests called chi-square to see that we're within the right ballpark. But for Biology 1, this is good enough, and we know that if we increase our sample size or increase the how well we count the corn, because sometimes the yellow looks like a purple when you're counting a whole bunch, we can get there. Looking up here, this was 2 classes worth of data. This was the sweet versus wrinkled, this was the purple versus yellow, and this was the dihybrids. Some of them were really close and some of them weren't, and so that's just a matter of how many ears were on their kernel and how accurate were their counts. But the one thing we did do was put all of them together. What we got was, when we put all the data together, we were 3.2 to 1 for the sweet versus wrinkled. Purple versus yellow would round up to 3 to 1, and the dihybrid would come really, really close. So all you have to do is finish answering the questions on the back of the lab sheet, and you've done the corn lab. Good job, Stu.
Refer to the illustration above. The child represented in box 1 in the Punnett square would A. Be homozygous for freckles, B. Have an extra freckles chromosome, C. Be heterozygous for freckles, or D. Not have freckles. The Punnett square in figure 11-5 shows that the gene for P shape and the gene for P color, A, assort independently, B, are linked, C, have the same alleles, or D, are always homozygous. The principles of probability can be used to A. Predict the traits of the offspring produced by genetic crosses. B. Determine the actual outcomes of genetic crosses. C. Predict the traits of the parents used in genetic crosses. Or D. Decide which organisms are best to use in genetic crosses.